Maybe not necessarily our airports now since COVID, but I still like to think of it as an airport. So we have these things right here. These are dendrites. So dendrites are going to be where information from around comes in. So this is going to be information from other neurons are going to talk to or enter in through here. So these are dendrites. So dendrites receive information or receive potential from other neurons and from the environment. That's what the dendrites do. So think of them like when you're driving up to the airport and you have like where you enter for Southwest and where you enter for Delta, right? You have all those different doors. That's these dendrites. And then you have to go and check in your bags, right? So this is a check-in bag area. We have our nucleus. We have our mitochondria. We have some cellular tissues, fibers. Right here we have our rough endoplasmic reticulum. Random thing about the rough endoplasmic reticulum and neurons, they're going to contain something known as Nissl substance. And Nissl substance is super important for protein development within neurons. And it's something exclusively found in the rough endoplasmic reticulum of neurons. So that's just a random trivia thing, which sometimes boards has random trivia things. So there's your little random trivia for the cell body. So just like at an airport, you check in your bags and then you're going to go to security. Security in the cell is this axon hillock. So think of this. I want to change the color I did this in because pink is disgusting. Let's do it in that color. So the security, think of this as like the security. They're going to choose whether or not they're going to allow you through. So at the axon hillock, this is where we have the decision, am I going to send the action potential down or not? Am I going to go through this process or not? So when we talk about action potential in a second, this is the area that's occurring. It's at this area that we're either going to activate or deactivate voltage-gated channels, those sodium voltage-gated channels. So this is going to decide action potential or not. That's what's happening here at the axon hillock. Neurons are like how I wish all people were. All or nothing. Either you give your all or you don't give nothing. There's no in the middle. There's no try, only do, right? So that's how axons work. So at this moment, it's whether is he going down to the terminal or not. So then you pass security. So say you get through the axon hillock and now you're going through the action potential. This action potential is going to occur right down here along this long axon we have. It's going to reach the axon terminal, which is where you're going to get off and get onto your airplane, which will take you wherever in the body you need to go. So that's your terminal, just like the terminal at the airport. So let's focus in what happens right here on this axon. What are these yellow things? That's going to be our myelin sheaths. And so our myelin sheaths are much like the, I don't know what the technical name, but they're like escalator floors. So, like as you're going through the airport, because people are so, lazy and they don't want to actually walk, they want to get places faster. They use the escalator floors instead of just walking next to them on like normal people have been doing for thousands of years. But we have escalator floors now. Well, in our neurons, we also have escalator floors, right? Those floors that move us or motorized floors. And those escalator floors go through saltatory conduction. So saltatory conduction is going to be the electrical event that occurs from node to node. So what we call the node is the area that is between, so right here, this is a node. It's called the node of Ranvier. I don't know who Ranvier is, but he is the one who discovered the node. And so what happens is, and then here's that myelin sheath I pointed out before. And so what happens during saltatory conduction is sodium, that's where the term salt comes in, right? Because sodium chloride is what is our salt, table salt that we have. So sodium, which I'm going to draw with this purple dot. So now I'm not going to write the word sodium again, but I'm just going to draw purple dots. So now you guys know. What happens is once I hit my action potential, I'm going to activate voltage-gated sodium channels. Okay, so right here you see it. This is my voltage-gated sodium channel. My action potential at the axon hillock, he gave the thumbs up. He was like, all right, ready for blast off. Go ahead, down my axon. So this opens up. And when it opens up, sodium is so excited. So damn excited. Sodium. So damn. I don't know if you guys see what I'm trying to do. I'm not doing it very well. But sodium is so excited to go in. So I remember sodium wants to go so damn in. It is, there's a lot on the outside, and naturally, sodium wants to go inside. Literally spends its whole life going, oh, I want to go inside the axon. Oh, I want to go on the inside the axon. So then this channel opens, finally the door opens, and just like a child at a fair, like it's almost Halloween, right? So just like those fun houses that aren't really that fun, that you go in as a child, you're like, oh man, I have to go in there. I have to go in the spooky house. I have to go in it. So you go in, but then just like children who go in spooky houses, they go, crap. There's no doors. There's no walls. There's no windows. Well, there's walls. There's no windows. It's dark in here. I don't like it. So then what sodium does is it rushes as quickly as it can until it gets to a place where it can go out. And so then it comes out again. But then it realizes, oh, I really so damn want to go in there. And so he goes back in. And he rushes and runs and sprints as fast as he freaking can to get through this hallway because it's scared. And then he's like, oh, but I really want to go in. And then that's what happens. So what we're going to see rhythmically, if I had to if you're a rhythm person, right? While we're at the node, we go kind of slow. But then we go basically 4 times when we're at the myelin. We're Speedy Gonzalez through that thing. Okay? And so what this does is we're going to propagate the signal. We're going to go faster through these myelin sheaths because there's no way out. And so we just want to get out as fast. The molecules get confused. They're like, oh, I don't like staying in one place for very long. Oh, let me go out. Oh, let me go out, right? They're coming in, going out. And so this is what happens during saltatory conduction. So some facts about the things going on here. My nodes, the job of my nodes. You're like, okay, well, Rhiannon, that's brilliant. Well, why not just make one long hallway that has no windows, that has no doors, and then the sodium will just go from the axon hillock all the way to the terminal. We don't need any nodes. It'll be super quick. Yeah, that sounds perfect. That sounds pretty awesome until you think about how much stamina do you think that sodium has? Well, let me tell you, those 5-year-old girls or boys who are going into spooky houses, if they couldn't find a way out, they probably, and I know this because I was one of them, will sit down in the middle of the house and cry and cry and cry until someone comes and finds them. And that's what sodium would do if we didn't have any nodes for them to get a breather, right? They would just give up and then they wouldn't reach the end. So the nodes are going to act to keep the signal going, to give them hope. You're our last hope, Obi-Wan, right? So that is what the nodes do. They keep the signals going. And then the myelin is there to propagate the signal. So that is saltatory conduction, and that is going to occur here down that length of that hallway that reaches the axon terminal. And so the axon terminal, which is kind of hidden over here. So we're going to put it down here, and we're going to change the color. Okay, the axon terminal there, that is going to be where we have a synapse. So this is the synaptic cleft. So sometimes you'll hear it called the synaptic cleft, the axon terminal. This is the end. This is where you get onto your airplane. Or I guess you go to another terminal. So how this works, so here we have our synapse. And action potential arrives. So it's traveling through saltatory conduction. It arrives here. And so action potential is a voltage. Okay? It's not like it's sodium runs. Sodium and potassium work to run this voltage. But it's not an actual molecule that's there or an enzyme or a protein there pushing along these little vesicles. So it's this voltage comes in, it comes down and it activates these voltage gated calcium channels. So actually I have this listed somewhere right here. Okay, yeah. So an action potential stimulates voltage activated calcium channels to open. Calcium is naturally found in between two neurons, two synapses, naturally. That is because calcium is found within all of our interstitium within our brain. All that cerebral spinal fluid interstitium stuff, yeah, we got a lot of calcium going on in there. When these channels open, calcium is going to rush into the axon terminal or into that synaptic cleft. So it comes in here, and calcium comes in, and then this calcium is like, yo man, let me push out the neurotransmitter for you, right? Because calcium, I think of like muscles and stuff. So I think of calcium comes in and muscles out the neurotransmitters. So as this comes in, it pushes the vesicles out, and these vesicles are full of neurotransmitters. What kind of neurotransmitter? We will talk more about what kind of neurotransmitters we find in a little bit. But they're going to push out neurotransmitters, and these neurotransmitters are going to go until they bind to the other side at the postsynaptic neuron. And so it says that here. So an influx of calcium causes release of neurotransmitters via regulated exocytosis. So it's exocytosis, right? It's exiting out of the nerve. What happens here? So we can have two different types of reactions at our postsynaptic neuron. The first thing is a ligand-gated ion channel. So that's kind of what I demonstrated in this image here. So remember that a ligand-gated